with the production of anti-inflammatories, antihistamines. The eosinophils also battle parasites by paralyzing them, and those include protozoans and helminths. And they do that by the production of antihelminthics and antiprotozoals in their granules. Okay, and then of course the neutrophils are there to attack and destroy bacteria. All right, now all of this is really like a first and second line of defense against infection. And then when we get to the last line of defense, that's your adaptive immunity, and that's your lymphocytes and your macrophages. And they base their attack on antigens, which are molecules that exist on the surface of the pathogen, all right, that are identifiers of the organism specifically, okay? And they use that as a way to identify and wipe out anything that antigen is attached to. And in the course of doing that, they actually develop memory cells that stay with you your entire life, okay? And that's why what doesn't kill you makes you stronger. Okay? I'm sure you've heard that phrase before, but in the immune system, that's actually true. Okay. So, immunity, right? First line of defense, a natural immunity. Okay, physical, mechanical, and biochemical barriers. Second line of defense, inflammation and the action of granulocytes. Third line of defense, lymphocytes and macrophages. Okay, so your physical barriers include your skin, okay, and your mucous membranes. Now, how is it that your skin protects you? All right, it does it several ways. You remember from A&P that your skin is an organ, right? And it's made up of different tissues. It's made up of connective tissue in the dermis and epithelial tissue in the surface. Hold on, we got some talking. We need to mute all here. There we go. There we are. Okay. So the surface is made of stratified squamous epithelial tissue that's keratinized, right? There's also in the dermis nerve endings and blood vessels and lots of white blood cells, okay? As well as lymphatic drainage and a lot of adipose tissue just beneath the dermis. That's the hypodermis, okay? So the surface of your skin is important in fighting off pathogens. Number one, because it's a dry, low pH environment, right? So you produce salts when you sweat, and oils keep your skin supple. And the bacteria that like to live in that produce acids that retard the growth of a lot of other bacteria that could be pathogenic. All right, so that's called the acid mantle. And in addition, right, the fact that you sweat and produce oils flushes the skin of stuff that would normally grow on it. Okay, so it's like hosing you off. Another important point is that the skin is constantly being shed, right? Here you go, sloughing off the cells, right? The reason that this is a big deal is because it gets rid of bacteria that want to grow on you before they really get a chance to start growing in any significant amount. So you shed off skin cells everywhere you go. Okay, everywhere you go, you leave a little bit of you behind. That's why 99% of dust is human skin. Okay. Just beneath the epithelial layer, you've got connective tissue. In the papillary dermis, you have areolar connective tissue. And beneath that, you've got dense, irregular connective tissue. And patrolling all that are white blood cells all over the place. And so anything that penetrates the epithelial layer, the white blood cells are laying in wait to pick off. So that's a big deal too, okay? The ultimate goal is to keep the pathogens from getting into the blood, okay? We call that septicemia if they're in the blood actively growing, and that's the worst possible outcome because that could kill you, okay? Other defenses, right? Coughing and sneezing to remove irritants that get in your respiratory tract, the flushing action of urine, vomiting to get rid of irritants in your GI, okay? And the fact that you have stomach acid, right? Which basically oxidizes, destroys anything you ingest, pretty much, not everything, but most stuff, is another protective measure, right? The upper respiratory has a mucociliary escalator, right? Pseudostratified ciliated columnar epithelia line this. Hold on. They line this, and there we go. They line this, and they produce mucus. The goblet cells make mucus, and that moves it up towards the oral cavity. And then we can either spit it out or swallow it. In any case, it doesn't end up in our lungs, which have mostly delicate, simple squamous epithelial tissue that needs to be well protected. Okay. The chemical barriers, right? Saliva, tears, earwax, sweat, mucus contain a lot of antimicrobial peptides like IgA and lysozyme, defensins, mannose binding lectin, proteins that bind iron and remove that from availability of bacteria to grow. Okay. Your normal microbes are a protection against infection as well. You've got a lot of guests in the hotel body, but most of them are good. And the good guests crowd out room for the bad guests to grow. And as a result, you're less likely to get infected with something that's a bad actor, a pathogen. Okay. So as long as your good bacteria and fungi are there, you're in reasonably good shape. Okay. So that's almost like another line of defense, really, that keeps you safe. Now, your second line of defense, inflammation. All right. Y'all know inflammation. Okay. The area that's inflamed gets red, swells up, it hurts. Okay. And it's warm to the touch. It's caused by infection, tissue necrosis, trauma, physical or chemical injury, foreign bodies. It's a product of chemicals that mobilize the immune system. Okay. And the point here is to bring blood to the area. And with the blood come the white blood cells, and with the white blood cells comes the ability to fight the pathogen that could get in through the injury or the area that's infected. Okay. The pain is there to indicate to you not to use the area that's inflamed because it needs time to heal. Okay. So pain is your friend. All right. With inflammation, we want to limit and control inflammation. We want to prevent and limit infection and further damage, initiate adaptive immunity, and initiate healing. So let's take a look at what inflammation is all about.
Although many things can trigger inflammation, our focus here is on how microbes cause the response. The process begins with the infection itself. Microorganisms trigger inflammation when they're introduced into the body and begin to grow and produce compounds that damage host cells. Resident immune cells, called macrophages, wander into the infected area and engulf and digest the invading organisms. They help to clean up the infection and, in the process, release inflammatory mediators, chemicals that call for more help. Some of these chemicals increase vascular permeability and stimulate vasodilation. Others are small proteins called cytokines that are made and secreted by cells as a way to communicate with other cells. Here they diffuse into the vasculature and stimulate endothelial cells to express specific receptors called selectins. Selectin binds to carbohydrates on the surface of neutrophils, snagging the cells as they flow by in the bloodstream and slowing them down, such that they roll along the endothelium. Inflammatory signals trigger these rolling neutrophils to express adhesion molecules called integrins on their surface. The integrins lock onto adhesion molecules called ICAM1 and VCAM1 on endothelial cells. The tight adhesion stops the rolling, and the neutrophils begin to stretch out along the endothelial surface. Damaged tissue cells in the area of inflammation release bradykinin, a 9-amino acid polypeptide that helps loosen the tight junctions between endothelial cells. Neutrophils can now initiate extravasation, in which they squeeze through the loosened endothelial wall and into the tissues, where they can help macrophages attack the invading microbes. Bradykinin molecules also bind to other cells of the immune system, called mast cells, that are in the area, causing them to release histamine. The histamine further loosens the endothelial cell junctions, allowing more fluid and cells to move out of the capillaries. Bradykinin, when it attaches to capillary cells, induces the cells to synthesize prostaglandins. Prostaglandins stimulate nerve endings, causing pain, which draws awareness to the infected area. The 5 cardinal signs of inflammation are redness, warmth, pain, swelling, and altered function at the affected site.
So it's for our own good, right? Eventually what happens is that we wipe out the invading pathogen and then we set the stage for tissue repair, okay? Other systems that aid us include the complement and the clotting systems and the kinin systems. Now, what we have to remember about all of these, right, is that they're all made by the liver, okay? Very important point here. Okay, so this is one of the reasons that people with liver damage, such as hepatitis, cirrhosis, okay, are impaired in all of these systems, right? They don't make sufficient complement, they don't make sufficient clotting factors, and they don't make the kinins. And what does that result in? Okay, well, and they also don't make sufficient albumin. Okay. Well, the complement system is important in helping to control infection, right? So they're more likely to get an infection, right? We'll see how complement works in just a bit. It gets its name from the fact that it complements the action of antibodies, all right? The clotting system, if you don't make sufficient clotting factors, that means you're going to bruise and bleed more easily, okay? So more easily infected. Okay, bruise and bleed. Okay, and then down here with the albumin, okay, they're going to have low blood pressure because they're going to lose fluid to other compartments like the abdomen. And one of the cardinal signs is ascites, right? Fluid in the abdomen, indicating that the lack of albumin, which is important in maintaining the fluid volume of the blood, causes the fluid to enter these other body cavities and the blood volume goes down and so the blood pressure goes down, right? So one of the things that you see in a lot of people with liver damage is that distended abdomen. Okay? So this is one of the reasons you got to treat your liver right, right? Because your liver does so much to keep you alive that if you're not careful, you're going to end up getting sicker quicker. Okay?
Was that referring to one of our diseases that we talked about?
Well, it's going to come back in a whole host of things, right? One of the things you're going to see with compromised liver function are all of these things as a cluster. Okay. So hepatitis and cirrhosis are 2 examples of conditions that affect the liver. Okay.
Trying to remember I'm trying to make a connection about what over the homework that we did. It was one of those, it was a giant it was over, you remember, it was like somebody had gigantism, like they're gigantic. There was another disorder after that. I don't remember the name.
With these 2, okay, hepatitis and cirrhosis.
Mhm.
Hepatitis is just a blanket term meaning that the liver is inflamed. Okay. It can be viral. It can be due to toxins. Okay. And it could be due to trauma, right? And it just compromises liver function. You get liver enzymes in your blood. Okay. Cirrhosis is a classic example of a type of hepatitis is caused by alcohol. All right. And what happens there with people that drink excessive alcohol is that when alcohol is metabolized, it actually turns into something similar to formaldehyde. Okay. And what that does is that kills liver tissue. And literally pickles the liver, right? Formaldehyde is a preservative. Okay. And acetaldehyde is an intermediate in alcohol metabolism that if it builds up to high amounts, it'll do the same thing to the liver, right? You know, the first four letters of the word liver, they spell live, and that's not an accident. That's on purpose. That was picked deliberately. Okay. So it's just a cautionary tale. A lot of things tax the liver, right? Prescription drugs, recreational drugs, alcohol, viruses, toxins, and stuff like that. All bad for your liver. Okay. So your liver is a huge MVP in keeping you alive. In addition, of course, it makes bile, which helps us to digest our dietary fat. Okay. And it helps to recycle iron in the body so that you can continue to make new blood. Okay. It also decontaminates the blood. It removes pathogens from the blood because there's little Kupffer cells that live in there, pick off pathogens in the blood that flows through it. All right. So that's like, but the nice thing about the liver, you know, I like to talk about the liver because, you know, it's underappreciated. The liver has immune privilege. Okay. That's one thing it's got. And it also regenerates.
There's something called cretinism. Is that in the liver or is that
You'd have to spell that for me. Can you spell that?
cretinism?
Okay. Cretinism, all right, is the result of a lack of thyroid hormone during
Okay.
development.
Oh,
Yeah.
okay.
All right. And what that does is that causes a whole cluster of symptoms, right? The child is short stature, has musculoskeletal problem, has cognitive deficit. Okay. Basically, the nervous system doesn't develop properly. All right. And unfortunately, there's no cure for that. Once you're born with a lack of thyroid hormone, there's no way to restore what's already been done, right? It's kind of like building a house. Okay. Building a house is a multi-step process. You have to buy the land and dig a hole, and then you have to pour a foundation, and you have to put up the frame of the house. And you have to put in the outside siding and the inside insulation, wiring, plumbing, and then the inner walls and the windows and the doors and the roof, right? And then you paint it inside and out. And then you turn on the gas and the electric and the water, and you have a house, right? If that stuff's not done properly, especially in the early first steps, everything else is going to suffer, right? For instance, if you screw up pouring the foundation on the first day, right? Crooked foundation leads to crooked frame, leads to crooked house, and probably eventually the whole thing falls over. And that's the same thing you're looking at here with cretinism, right? Cretinism is something that is during development, right? The thyroid hormone has to be produced by the fetus, all right, in order to support development of the nervous system, right? He's not getting it from the mother. He's getting it primarily from his own production. And that means that the nervous system doesn't get built properly, as well as these other problems that are system wide. Okay. And so that's the unfortunate outcome of that. Okay. But back to your liver, right? Immune privilege and regeneration, right? The regeneration liver is impressive, right? You can lose 80% of your liver and it'll grow back. Okay. The immune privilege just means that you can take a liver or a liver lobe and you can transplant it into somebody else, and as long as the blood type matches, that tissue will take. Okay, so it's not nearly as restrictive as organ transplants for things like a kidney or a lung lobe or something like that. Right. So those are two benefits of the liver that, you know, I guess our creator provided us because they knew that he or she knew that we would need it. Okay, so important. I just, you know, always pause when the liver gets involved because it's a big deal. Okay. The plasma protein systems are made up of inactive enzymes in the plasma made by the liver that get activated in what's called a cascade. Okay, cascade. Okay, cascades you've seen in A&P because they are inside and outside the cell, right? The clotting cascade is something that we'll talk about. That's an extracellular cascade. Okay. Signal cascades are something that's inside the cell. All right. And all you have like an initial event. Okay. And that leads to another secondary event, all right, that triggers an inactive to an active form. Okay. And that leads to another event that goes from an inactive to an active form. All right. And then and eventually you get to the response. All right. Now, why is that a big deal? Because it takes a small event and then at every step, you magnify it geometrically because these next events can happen multiple times, right? And as a result, the response grows at each step and becomes more effective. Okay. Those geometric progression gets bigger and bigger and bigger and bigger. And you get the response. And then it's easy to turn off because all you have to do is block like the first event. And then the cascade will stop. And then those activated molecules will get deactivated by enzymes that are hanging around to do that. And everything goes back to where it was. Okay. So it's a theme in biology, the cascade signaling system. Okay. The first proenzyme gets converted to an active enzyme. The substrate of the activated enzyme becomes the next component, and cascade, cascade, cascade. Okay. The complement system makes active fragments that recruit phagocytes, activate mast cells, and destroy pathogens. Activation of C3 and C5, as we'll see, results in opsonization, chemotaxis for white blood cells, anaphylactoxins. The pathways include the classical, the lectin, and the alternative pathways. So let's look at complement here.
The complement system consists of a group of over 30 serum proteins that play an important role in both innate and adaptive immunity. Complement proteins participate in host defense processes such as inflammation, attraction of phagocytes to an infected area, enhanced effectiveness of phagocytes, and lysis of cells. Complement proteins are activated by a cascading mechanism. There are 3 pathways of complement activation: the classical pathway, the alternative pathway, and the lectin pathway. The classical and alternative pathways are demonstrated in this animation. The classical pathway requires interaction of antibodies with an antigen, usually on a cell surface. Following binding of antibodies to cell surface antigens, C1, which is composed of 3 proteins, R, Q, and S, attaches to the constant region, Fc, of adjacent antibodies through its C1q subunit. C1s then acts on C2, cleaving it into C2a and C2b. Then C1s cleaves C4 into C4a and C4b. C2b and C4b bind to the cell near the antigen-antibody complement complex, and C2a and C4a are released. This complex of C4b and C2b, called C3 convertase, has protease activity and cleaves C3 into C3a and C3b. C3b becomes bound to the cell surface, whereas C3a is released. C3b then absorbs to C4b2b to form C4b2b3b. This structure is called C5 convertase. The alternative pathway does not require the presence of antibodies. Activation begins with cleavage of C3 into C3a and C3b, either spontaneously or by a blood protein. C3b binds to foreign substances. Factor B, a blood protein, attaches to C3b and is cleaved into two fragments, BA and BB, by factor D, forming C3BBB, analogous to C4B2B. Both structures, C4B2B and C3BBB, are called C3 convertase. C3 convertase rapidly cleaves more C3 into C3a and C3b. C3 convertase is further stabilized by properdine, which allows another addition of C3b, forming C5 convertase.
So a cascade, a series of events that leads to activated complement, right? So how is it then that this actually ends up battling infection, right? Opsonization is the coating of bacteria with proteins that make it easier to engulf by a macrophage, okay? So it improves the grip, right? Chemotactic factors attract white blood cells, okay, to do battle, right? Macrophages and other types of white blood cells that have phagocytic activity, all right? Anaphylotoxins, okay, enhance inflammation. But also induce cell death in the pathogen. Okay. So all of that, right? A cluster of attacks. So you can see why if you lack complement, you're going to have trouble clearing infections because it's like your backup doesn't show up to help you out. Right. Now the clotting system is there to prevent hemorrhaging, which is a fancy word for bleeding. Okay. Forms a fibrous meshwork at an injured or an inflamed site, prevents the spread of infection, localizes microbes in foreign bodies, forms a clot that stops bleeding, and provides a framework for repair. Main substance is an insoluble protein fibrin, and there's two pathways, the extrinsic and the intrinsic pathway. So let's look at them.
At the site of vessel injury, the first platelets arrive to start sealing the wound. Simultaneously, the coagulation cascade with its various coagulation factors is activated. This involves two pathways, the extrinsic and the intrinsic pathway. Extrinsic activation begins with now exposed molecules of the vessel wall, such as tissue factor, which forms a complex with factor 7. Finally leading to the activation of factor 10. This factor 10A is the point at which the extrinsic and the intrinsic pathways of the coagulation cascade meet. The intrinsic pathway consists of various coagulation factors activating each other in a chain reaction. At its end, a complex with an additional cofactor is formed. This complex now activates factor 10. Since the two pathways merge at the level of factor 10A, this factor has a pivotal role in the coagulation cascade. Further down the cascade, factor 10A, in combination with 5A, activates thrombin and induces the so-called thrombin burst. one molecule of factor 10A can catalyze the formation of 1,000 molecules of thrombin. These large amounts of thrombin cause the further activation of platelets and the enhanced formation of fibrin. Fibrin then forms strands, making up the mesh that stabilizes the platelet plug in an arterial clot and holds together the red blood cells in a venous clot. It can be concluded that the central role of factor 10A in the coagulation cascade makes it a viable target for therapeutic intervention in pathologically altered blood coagulation.
So again, right, this is part of a process called hemostasis. Okay. Which is just fancy for cessation of bleeding. Stop bleeding from bleeding. Okay. So you get an injury, okay? You get vascular spasm. Okay. That slows flow. And then we initiate clotting. Okay. And then what's going to happen, right, is that prothrombin is going to be converted to thrombin, all right? And then fibrinogen, which is another plasma protein, is going to be converted into fibrin, and that's your clot, right? And that's going to block the opening so that the bleeding will stop, and it's going to trap stuff in it, okay? And then you're going to get retraction in the clot to draw the wound more closed, okay? And then you're going to set the stage for tissue repair underneath what will be the clot. And then eventually, once that happens, the clot gets dissolved and is no longer necessary, right? So the problem, of course, if you have liver damage, is that you're going to bleed more easily because you're not going to be able to set up clots, right? And that's going to mean that it takes longer to repair an injury. And it's also going to help spread infection. If you have infection, it goes with that as well, right? So this is a huge problem. There are people that are born without some of the clotting factors, okay? And that's a condition you may have heard of called hemophilia. Okay. And if you have hemophilia, you're what's called a bleeder. You lack one of the components of this cascade, and so you don't make the clot. And so you just continue to bleed, right? It affects men more than women because the gene is on the X chromosome. And of course, guys only have one of those, okay? It's a recessive disorder. So you only need one copy in a male for it to show up. And the fix is to give the clotting factor that's missing in the affected male. So the way that this disease generally works is he gets it from his mother who carries it, okay? Any of those clotting factors that are missing, you're going to be a bleeder of some kind, okay? Women can be hemophiliacs, but it's very, very rare because they would have to have two recessive copies, one from their mother and their father, in order to be bleeders. Okay, the kinin system functions to activate inflammatory cells. Bradykinin is one of the main factors. It causes dilation of blood vessels, pain, smooth muscle contraction, increases vascular permeability. Basically, aids in inflammation, okay, which we've already heard about, right? Helps to control the interaction of plasma proteins. Tight regulation is critical. A lot of mechanisms available to activate or inactivate those plasma protein systems after they've executed, right? The components of inflammation, erythrocytes, platelets, leukocytes, okay, which include the granulocytes, the monocytes, the lymphocytes. So the let's go through these for a minute here. There we go. All right. So the granulocytes, right, are the eosinophils, the basophils, right, and the neutrophils. Okay. The monocytes become macrophages in the blood. And of course, the lymphocytes are our adaptive immunity. Hold on. And our adaptive immunity is our B and T lymphocytes. Shh.
Blood is responsible for transporting oxygen to the cells and removing carbon dioxide for excretion by the lungs. Blood carries nutrients, ions, water, hormones, and enzymes to all body cells and carries away the waste products. Blood regulates the pH and temperature of the body. Blood is composed of the liquid portion, plasma, and solids called formed elements. Plasma makes up 55% of the total blood volume, 91% which is water. Plasma proteins are albumin to maintain osmotic pressure and water balance, globulin antibodies to help the immune system, and fibrinogen, an essential component for blood clotting. The formed elements are erythrocytes or red blood cells, leukocytes or white blood cells, and thrombocytes or platelets. Red blood cells have no nucleus, therefore they do not divide and only live 120 days. Red blood cells contain hemoglobin that carries oxygen to all cells of the body and then carries away carbon dioxide. There are 5 types of white blood cells, 3 granular and 2 non-granular. Under the microscope, the granular types appear to have brains. They are neutrophils, eosinophils, basophils. Neutrophils phagocytize or eat up foreign substance and secrete lysosomes that destroy bacteria. Eosinophils produce antihistamines that combat irritants. Basophils are also involved in allergic reactions, secreting heparin, histamines, and serotonin. Monocytes also phagocytize bacteria and dead cells, and when they enter the tissue, they are called macrophages. Lymphocytes produce antibodies and are crucial to the body's immune system. Bacteria are being destroyed by neutrophils, lymphocytes, and macrophages. Platelets initiate the chain reaction in clot formation.
Cellular receptors include PRRs, and the antigens include PAMPs. So the PRRs bind the PAMPs. Damage-associated molecular patterns, toll-like receptors, complement receptors, scavenger receptors, and knot-like receptors. The idea with this is that when receptor meets its target, it triggers a response in the white blood cell that's going to result in an attack, okay, so that we can wipe out the invader. All right, so this is sort of, it's similar to adaptive immunity, but not entirely. These molecules, these PAMPs, are sort of common to a broad number of pathogens, okay, and not a specific antigen on a particular strain of, say, invading pathogen. Right? Cytokines are basically mediating the inflammatory response. They're made in a lot of different cells in response to pro-inflammatory cytokines. They induce chemotaxis. Now, what is that? Chemotaxis is the ability to move towards a chemical signal or away from a chemical signal. In this case, we're talking about positive chemotaxis. We move towards a particular chemical signal. Okay, it promotes phagocytosis and wound healing. Examples include macrophage chemotactic protein, macrophage inflammatory protein, and neutrophils. Okay, all of these guys, the macrophages and neutrophils are all eaters. Okay, they come in and they eat the bad guy, and they eat dead tissue and process it and pitch out the waste products. Okay, interleukin literally means between white blood cells. Okay, they're made by macrophages and lymphocytes in response to PRRs, okay, or other cytokines. Examples include IL-1, which is a pro-inflammatory, and IL-10, which is an anti-inflammatory. Okay, they're going to have a role as well in adaptive immunity that we'll see later. Okay, TNF alpha is secreted by macrophages in response to PAMP and toll-like receptor. It induces fever by acting as an endogenous pyrogen. It increases synthesis of inflammatory serum protein and causes muscle wasting and intravascular thrombosis. Now, important point here with the fever, right? Pyrogen means fire, okay? Or high temperature, if you like. A pyromaniac is somebody that sets fires. Okay? The idea here is to elevate the body temperature, all right, to a point where it's good for our white blood cells, but bad for the pathogen. We try to cook him out so that he can't do his damage the way he likes. Okay, so that's why fever, and that's why inflammation with the elevated temperature near the site. Okay, interferon protects against viral infections. It's produced and released by virally infected host cells in response to double-stranded RNA and protects the nearby healthy cells. Interferon alpha and beta cause production of antiviral proteins. Interferon gamma increases microbiocidal activity of macrophages. It also is an anti-cancer. Modulator. Okay, because it jacks up natural killer cells activity. Okay, so how exactly does this work? Well, I'll give you one example, right? You get invaded by a virus, you're compromised, all right? You're not going to make it, but in response to double-stranded RNA, you kick out interferon that binds to neighboring cell receptors and induces the production of something called dicer. Okay, well, what does dicer do? Dicer chops up double-stranded RNA. Okay, and so what happens when that virus comes into that cell that's been alerted by interferon is it doesn't have any chance to complete its replication cycle, because the minute it produces double-stranded RNA, it gets split to bits, okay, and it can't complete. So everybody was very excited about interferon. They thought, oh my God, this is a miracle drug, right? IFN. Let's start making IFN and giving it to people because it's an antiviral and an anti-cancer drug, okay? And they did that. And then in clinical trials, what they found out was that high levels of IFN are hepatotoxic. Okay, meaning what? They do a number on your liver, all right? So there's a reason the body makes this in small amounts, all right? And that turned out to be the reason, right? So we still give it, but we give it in low dose. And it's only going to be effective against RNA viruses, all right? Those that do their dirty work entirely in the cytoplasm, with the production of double-stranded RNA being part of their replication cycle, right? It's not really going to help us against DNA viruses like the herpes class, because they get down there in the nucleus and they either live there or integrate there, and they become a permanent resident, right? You can never get rid of them. But against the RNA viruses, it is beneficial, okay? High levels can be lethal. Well, we just talked about why, right? All right, cool. Mast cells and basophils, they're really two ends of the same thing, right? A mast cell is like a fixed basophil, and a basophil can wander around. And what they do is they produce massive amounts of heparin and histamine, okay? And of course, the histamine initiates inflammation, all right? And the heparin slows blood clotting, all right? They also produce chemotactic factors to attract white blood cells, cytokines to activate them, okay? The chemical release is through degranulation, right? Where basically they spray, all right? New production and release of mediators in response to a stimulus is synthesis, right? So you make more and you spit out more, right? So what happens when this occurs, right, is that you cause dilation of capillaries in response to the histamine, and that's going to result in blood infusing into the area, and with that, the white blood cells. Okay, you've got eosinophil and neutrophil chemotactic factors that bring those guys in. All right, the vascular effects of platelet activating factor. Right, platelet gets activated because of synthesis of phospholipase. All right, and the arachidonic acid from that causes production of prostaglandins and leukotrienes, right? So, we hurt, our capillary beds become leakier, the blood gets into the area, and with that, the white blood cells, and with that, the healing, okay? The pain we've talked about, right, as a benefit, okay? The attraction of the white blood cells to battle the pathogen, okay? But what happens when this goes haywire, all right, is that we open up all our capillary beds at the same time, all right? And if we open up all our capillary beds at the same time, our blood pressure can drop to the point where we could go into a coma and die, all right? Examples of that include toxic shock and septic shock, right? Okay. It's the same mechanism in each scenario, right? And toxic shock is something that's in your blood that triggers the mast cell release, and then everything goes haywire. Okay, this can be mediated by IgE, okay, antibody that binds the trigger molecule and then sits on the mast cell, and when it does that, it releases all of this histamine and heparin and all these other components, right? When all your capillary beds open up, your blood pressure drops dramatically, because at any moment in time, normally 2/3 of your capillary beds are closed. That's normally, okay? But with this stuff, they all open up, all right? And your blood pressure drops big time, okay? And septic shock is just a response to the antigens that are on the bacteria that are growing in the blood, okay? And so that's something that we don't want on a large scale, okay? We want this locally, but we don't want this all over the body. Okay? The receptors, H1 receptor is pro-inflammatory and smooth muscle cells of the bronchi, okay? And H2 receptor is an anti-inflammatory, present on parietal cells of the stomach, and induces secretion of gastric acid, okay? So this is one of the reasons that you get bronchoconstriction. and mucus production. With a pro-inflammatory response. People that have asthma, all right, have this to a high degree, all right. And so what happens is they get that massive histamine release. The H1 receptors bind the histamine, and then they get so bronchoconstricted and clogged with mucus that they can't breathe. Okay, so generally, with an asthmatic, okay, you carry an inhaler either of epinephrine or albuterol. I think that's the way it's spelled. It's ER. Okay. And those are bronchodilators. Okay. Neutrophil chemotactic factor induces, attracts neutrophils. Eosinophil chemotactic factor in anaphylaxis attracts eosinophils. Okay, so the idea here is draw them to the area so that they can do their work. Degranulation is just basically the granulocyte is spraying. Okay, he's spraying out his stuff. Okay. Mediators, leukotrienes are made from arachidonic acid from mast cell membranes, similar effects to histamine in later stages. Prostaglandins are similar to leukotrienes and induce pain. Aspirin and nonsteroidal anti-inflammatories, NSAIDs, block the synthesis of prostaglandins and inhibit inflammation and pain. Okay, platelet activating factor is similar to leukotrienes and platelet activating. Okay, so the platelet activating factor is again to promote clotting. Okay, these prostaglandins, right? The pain, remember, is an indication not to use the area while it heals. Okay. And again, the leukotrienes are promoting the leakiness in the vessels. Okay. The NSAIDs is an interesting thing, right? Nonsteroidal anti-inflammatories are given to block synthesis of prostaglandins and inhibit inflammation and pain. Yay, you feel better, right? However, by feeling better, what are you doing? You're blocking the synthesis of prostaglandins and inhibiting inflammation and pain, okay? Which means what? You're going to be less effective at battling the infectious agent, okay? So you have a choice, right? You can either feel really unwell for a short period of time, or you can feel less unwell for a longer period of time. The choice is up to you, okay? Now, NSAIDs imply that there are SAIDs, right? Non-steroidal anti-inflammatories imply that there are steroidal anti-inflammatory drugs, and there certainly are, and those are usually glucocorticoids. Right? Well, so why don't we just use glucocorticoids for everything? Well, because they're going to suppress, not only suppress inflammation, okay, but they also increase blood sugar levels, right? And if we do that for long periods of time, number one, you're going to be hyperglycemic. Number two, you're going to get sicker quicker because your inflammatory process is going to be suppressed so much that you're going to leave the door open for infection, right? And this is because of the effect of the glucocorticoid, right? The glucocorticoid does that. It suppresses inflammation and increases your blood sugar. It's very effective. Okay, it knocks inflammation down immediately. But what you could end up doing is something that we kind of talked about in the last chapter. You could induce Cushing's disease. Which is basically an overproduction of cortisol, right? And in this case, if it's because of a treatment, we call it iatrogenic. Iatrogenic Cushing's disease, meaning Cushing's disease brought on because of the treatment. Okay, so we try to avoid that and use the NSAIDs instead of the SAIDs whenever we possibly can. Okay, all right. Endothelia adheres to underlying connective tissue, and that forms basically the capillary layer. All right, it's just endothelial tissue, basement membrane, and basically that's it. Okay. It interacts with circulating cells, platelets, plasma proteins. It regulates circulating inflammatory components. Damage to these initiates the platelets sticking. Okay, so generally, the endothelial cells are coated with something called prostacyclin. Okay, which you could kind of think of as Rain-X for the blood vessels. Makes them slickery, okay? And so formed elements don't stick, all right? But you damage that, what do? You expose collagen fibers and other stuff, and then the platelets are going to stick, and they're going to release factors that attract more platelets, and then you're going to eventually make a platelet plug, right? So tissue destruction and inflammation leads to interaction with coagulation cascade to stop the bleeding. Degranulation and serotonin acts like a histamine, okay? And then we start the production of the platelet plug, attract more platelets, stop the bleeding, and then we're going to set up a blood clot, all right? And then tissue repair under that, and then eventually the clot will be dissolved, okay? Neutrophils are also called polys, okay, because they have nuclei with 3 or more lobes, and they got a variety of shapes. They're important early in inflammation because they battle bacteria. Okay, that's their job, is to eat and kill bacteria. They're short-lived. They get extruded as pus. Okay, and we should talk a little bit briefly about why they have their names, right? Eosinophils, basophils, neutrophils, right? When you do a blood smear, you stain it with something called right stain, okay? And right stain consists of a red dye called eosin and a blue dye called methylene blue. Okay. And the idea behind these names is phil means to attract or to like, right? So the eosinophils granules are attracted to the red dye eosin. That's why they look like they have red granules in them under a microscope. The basophils are attracted to the basic dye methylene blue, so their granules will be purple, all right? And the neutrophil granules are attracted to each of the dyes, and so their granules look pink, all right? So that's where the name came from, right? Just so you know. The eosinophils are sort of like the other end of inflammation, right? They're phagocytes. They defend against parasites and regulate vascular mediators. They produce antihistamines. Okay, so they help to turn off inflammation, right? So they're the other end of it. The basophils, again, are the inducers of inflammation, right? Monocytes become macrophages when they enter the blood, and their job is to eat the invaders, all right, and kill them as a result, right? But the other thing they do, right, is they are antigen presenting cells, APCs. We're going to find out in adaptive immunity that that's the part where a little piece of the pathogen gets shown to the lymphocyte, and as a result, the lymphocyte gets activated and is ready now to clone himself and make a little army that goes out and kills whatever the antigen is attached to. In the case of the B lymphocyte, the weapon is the antibody. In the case of the cytotoxic T lymphocyte, they show up at the infected cell and tissue and destroy it. Okay. Dendritic cells are in peripheral organs and skin, and they migrate through lymph vessels to lymph tissue and interact with T lymphocytes to generate acquired immunity. Okay, so we would call these guys, right, wandering macrophages. Okay, and dendritic cells are what we would call fixed macrophages, meaning they don't leave their tissue, they stay where they're supposed to be. Well, these guys wander around through the blood. Phagocytosis is just a way to eat the invader, right? Production of adhesion molecules, margination, right, which is the sticking of the leukocyte to the endothelial cell to get to the bad guy. Diapedesis, we squeeze through those little clefts, and then positive chemotaxis draws us to the invader, right? So adherence, engulfment, phagolysosome formation, okay, and destruction of the target, right? So we have an injury. With the injury comes the pathogen. The damaged tissue releases chemical signals. The chemical signals cause vascular permeability and the production of molecules on the endothelia that allow those cells to stick. Okay, that's the margination. The vascular permeability lets the cells squeeze through the clefts and then march towards the pathogen and eat him up, kill him. Okay, you can see here, right, that you can have a variety of ways to grip your guy, recognition and attachment. Okay, you see that antibodies and the use of opsins are a part of this, right? C3b, right? Makes him easier to grip and destroy, right? We pull him inside, engulf him. The lysosome fuses with the phagosome and chews him into little bits, and he's dead. But we go a step further and we present pieces of him on the surface to alert our adaptive immunity. Okay, so you can see in the diagram here, right? This is complement doing its work. Okay, antibodies, which are from adaptive immunity. Okay, the antigen is from the pathogen. Okay, so this is what's going to happen in the next chapter, chapter 7. We'll talk about antigen antibody as part of B cell activity. Okay, and then here we go with these PAMPs and PRRs, right? Which are just a basic way to grip you, because you know you don't have a PAMP if you're a normal cell, right? So it's a form of recognition of an antigen, but it's different from our adaptive immunity. Okay, so we pull all that in, and then we got it. Okay, natural killers recognize and eliminate cells infected with virus. Inhibitory and activating receptors allow differentiation between normal and abnormal cells. They make cytokines and toxic molecules, and also, more importantly, they destroy cancer cells. They do this every day. Okay, they punch holes in them and kill them. Now you might say, well, so what? Compared to infection, that's not a big deal. But believe it or not, everybody in this class gets cancer 2 or 3 times a day because of mutation. All right. And these natural killer cells are important in destroying those cancer cells before they get a chance to spread. That's called metastasis. All right. And that is a function called surveillance. Okay. Surveillance. Okay. That's why people with suppressed immunity have higher cancer rates. Acute and chronic inflammation. Well, acute is self-limiting, results from vascular changes and leakage of circulating components into the tissue, heat, swelling, redness, pain, like we've already talked about, right? And fluids, right? Okay. The fluids includes a serous exudate, which is watery and indicates early inflammation. Fibrinous exudate, which is thick clotted exudate, indicates advanced inflammation. And purulent exudate means there's dead neutrophils in there, which means you're doing battle with the infection. That's pus. Okay, so bacterial infection's going on. Hemorrhagic exudate contains blood and indicates you're bleeding. Okay, fever, right, caused by exogenous and endogenous pyrogens acting on the hypothalamus. Leukocytosis, you're going to see an increase in your white blood cell count with an infection. That's what we would expect. Okay. Increased plasma protein synthesis includes C-reactive protein, fibrinogen, haptoglobin, amyloid, and ceruloplasmin. Okay. And all of this is designed to deal with the infection. Okay. We talked about the function of the fever, the jacked white count. It makes sense, right? Because you want more white blood cells to deal with the infection. Okay. And the plasma proteins aid in that, all of those processes. Okay. With chronic inflammation, it's lasting 2 weeks or more. It's related to an unsuccessful acute inflammatory response. We see pus formation, suppurating wounds, and incomplete wound healing. Causes high lipid and wax content in the microbe. Okay, that's one of the virulence factors that certain microbes have. A virulence factor is a weapon that helps the microbe produce disease. The ability to survive inside a macrophage, another trick that's used by some pathogens to avoid destruction. Toxins and chemicals, particulate matter, or physical irritants, right? So you can see here what's going on with chronic inflammation, okay? So we have the persistence of an infection by an antigen or a foreign body, okay? We have neutrophil degranulation and death, lymphocyte activation, fibroblast activation, okay? Lymphocytes and monocytes and macrophages infiltrate and make more pus. And eventually we may get to tissue repair with a scar. Okay? Characteristics: dense infiltration with lymphocytes and macrophages, granuloma formation, epithelioid cell formation, giant cell formation. Okay. Wound healing is one of those things that happens in tissues, and it varies according to the tissue that we're talking about. Okay. Regeneration is something that's possible in epithelial and connective tissue. Okay. Not in muscle tissue and not in the neurons of nerve tissue. Okay. Resolution, returning the injured tissue to its original structure, repair, replacement of destroyed tissue with scar tissue. Okay. So what sort of tissue? Scar. Let's talk about that. Muscle tissue, nerve tissue. Okay. We were talking about mostly the neurons. Okay. And then in epithelial tissue, we get scar tissue if the stem cell population is destroyed. Okay. Okay. such as a deep cut in the skin, right? You get a deep cut in the skin, you don't get full function back, right? Scars are composed mostly of collagen to restore strength in the tissue and not necessarily full function, right? We fill in the wound, we seal it, and we shrink it, okay? An example, right? A burn victim. Burn victim is going to have scarring as a way to patch the wounded areas, but they're not going to have the full ability of the skin to do thermoregulation, to produce oils and sweat. There's not going to be any hair follicles, all right? All of that function is gone, okay? Which is why extensive scarring compromises the normal role of the skin. Now, you can also scar inside your body. You can scar your lungs, you can scar your liver, okay? You can scar your GI, all right? It's all the same deal, right? And when you scar, you lose the function of that tissue, okay, where the scar formed. Primary intention, wounds that heal under conditions of minimal tissue loss. Secondary intention, wounds that require more tissue replacement, such as an open wound, okay? The inflammation phase, we see coagulation, infiltration of wound healing cells, and then in angiogenesis, we grow new blood vessels, okay? The proliferative phase sees granulation, epithelialization, and requires fibroblast proliferation, collagen formation, and wound contraction. Why? We have to have a framework upon which to build everything else, okay? So we can see here what's going on, right? With remodeling and maturation, we continue cellular differentiation, we have scar tissue formation, scar remodeling. So see how it takes place, right? We have the initial injury, and then we bleed into the injury, we hemorrhage, right? And then we see platelet plug formation. Neutrophils come in to clean out dead tissue and pathogen, okay? And then in a couple of weeks, we see proliferation. Mitosis is triggered, granulation, tissue forms, okay? Lymphocytes are still present. Fibroblasts are laying down new matrix, new capillaries grow, okay? And in weeks to months, you get the fibrous union and the remodeling, all right? Now, in secondary intention, right, we see a wound, we see the proliferative response, we see wound contraction, okay? This is a more extensive injury, right? If we go through the stem cell population, as we've done in each of these cases, we don't get full tissue function back, okay? Dysfunctional wound healing can occur in any phase of wound healing. Examples include ischemia, excessive bleeding, excessive fibrin deposition, predisposing disorders, diabetes, obesity, infection of the wound, inadequate nutrients, drugs, and tobacco smoke, right? Dysfunction during reconstruction, you can end up with dysfunctional collagen synthesis, such as a keloid or hypertrophic scar, okay? Wound disruption, which is dehiscence, which increases the risk of infection. So the wound pops open again. Impaired contraction, which generates contracture. So you pull too much on the wound and you generate now less tissue around the area than there was before and reduced mobility, right? So you can see some examples here, right? You can see keloid here. There's overscarring basically. And there's a major genetic component to that, right? Okay. Let's take 15 minutes and get into the next chapter. Start these. Let's pause the recording. I just want to pause it.